So it is a pleasure for us to have today in our Byline Vision Science Seminar, Professor Rachel Denison from Boston University. Professor Denison completed her BA in Cognitive Science with distinction at Yale, then completed her master's degree in neuroscience at Oxford University, and then completed a PhD in neuroscience with a designated emphasis on computational science and engineering at UC Berkeley at the Helen Wales Neuroscience Institute, and this was done under the supervision of Professor Michael Silver. She then continued to do a postdoc at the Department of Psychology and Center for Neuroscience in NYU, working with Professors Marisa Carrasco and David Heeger. During her pre-faculty career, she also worked with Professor John Driver, may his soul rest in peace, and with Professor Marvin Chun. After that, she became faculty at the Department of Psychological and Brain Sciences in Boston University, where she's now affiliated with the Cognitive Neuroimaging Center, the Center for Systems Neuroscience, and the graduate program in neuroscience at Boston University. Throughout her career, she has received multiple awards and distinctions. Her research is consistently funded by multiple distinguished funding bodies, including NSF, NIH, and the Templeton Foundation, where she collaborates with groups of highly acclaimed researchers to investigate and address profound questions in neuroscience and human being. She publishes in top leading journals as Behavioral and Brain Sciences, Nature Human Behavior, PNAS, Nature Reviews in Psychology, and other journals, and she also took part in writing 2 book chapters. She's invited to dozens of talks, and she also takes part in scientific outreach activities. Her lab studies visual perception, attention, and decision-making with a focus on temporal dynamics while integrating behavioral measurements as psychophysics and eye tracking, neural measurements as fMRI, EEG, MEG, and computational modeling. While there is so much more to say, I'll stop here and welcome you, Rachel, to our seminar to hear about your exciting research, and thank you so much for joining us today.
Well, thank you so much. Thanks for the introduction, and thanks for the invitation. I'm really excited to be here and share this work with everyone. Okay, so let's get started. I mean, this is a view right here from someone biking through Midtown Manhattan, not me. And this gives us a sense of really what the visual system has to process. It helps us appreciate that our environment is highly dynamic. We have to perceive and act in real time based on this constantly changing stream of visual input. Now, attention is a cognitive process that lets us prioritize relevant visual information. And it's important because we have limited neural resources with which to process all the information that's entering our eyes. One very well-studied type of attention is spatial attention. For example, even if you're keeping your eyes straight ahead on the road in front of you, you might attend to the right because you want to change lanes and you want to make sure that no cars are coming. Now, a hallmark of spatial attention is that it is selective. So selective means that if you attend to the right, you see better on the right, but at the cost of seeing worse on the left, which you've been ignoring. Now, today I'll be talking about temporal attention. So we can attend to specific points in time when something important is likely to occur. For example, this biker driving down this busy street should attend more at this time, right, to see what this blue van is doing and avoid running into it right now. Yeah. So that's temporal attention, which is the prioritization of visual information at specific points in time. And this kind of goal-directed attention to a point in time that you know in advance will be relevant for your task goals is called voluntary temporal attention. We've used an integrated approach to study voluntary temporal attention in which we characterize behavior, develop computational models to make our working hypotheses explicit, and then use these models to make new predictions about brain activity, which we then test with experiments. And I'll touch on all of these points today, but I'll focus mostly on behavior and neural mechanisms. Now, although temporal attention is a growing field of research, we still know much less about voluntary temporal attention than we do about other types of attention. And really, time is a very different kind of dimension than space or features as far as the brain is concerned. It's still unclear whether temporal attention and other types of attention have common or distinct mechanisms. So the first question we asked when we were getting started with this research on temporal attention was, is voluntary temporal attention selective in time, just as spatial attention is in space? So selectivity in time implies trade-offs, here not across space, but across time. And that looks like this. So if you have two stimuli coming one after the other in a sequence, let's call them T1 and T2. And if you know in advance that T1 may be relevant for your task, you could attend to T1 and experience an attentional benefit for T1. But if attention is selective in time, this should come at a cost later in time at T2. And vice versa. If you attend to T2, you may have an attentional benefit for T2, but if attention is selective in time, this should now come at a cost, and now this would be earlier in time at T1. And this is what we call the perceptual trade-offs hypothesis. Alternatively, if there are only two stimuli, one after the other in the same spatial location, why can't you just sustain attention at a maximum level for both? Then attending to one point in time doesn't mean there's less attention available at the other point in time. And this is what we call the sustained attention hypothesis. So to test for selectivity, we developed a new task, a two-target temporal pre-cueing task, in which participants are instructed in advance to attend to a particular time point. So on each trial of this task, we have two targets presented sequentially in the same location, T1 and T2, separated by this short interval. And the observer's task is to discriminate the orientation, clockwise or counterclockwise, of one of these gradings, which is indicated by a response cue at the end of the trial. We manipulate temporal attention trial by trial using a pre-cue at the beginning of each trial, which instructs observers to attend to T1, attend to T2, or attend to both targets in the neutral condition. Now, this pre-cue is 75% valid, meaning that 75% of the time, the response cue matches the pre-cue, and these are the valid attention trials. The other 25% of the time, the response cue mismatches the pre-cue, and these are the invalid attention trials. And then in neutral trials, the pre-cue is uninformative, and the response cue is equally likely to probe each stimulus. So observers should attend to both. Behaviorally, we find that temporal attention improves discrimination of the orientation in this task. So we found for T1 that accuracy was highest for valid, intermediate for neutral, and lowest for invalid trials. We see the same pattern for T2. And for both targets together, we see significant benefits of temporal attention in valid trials, as well as costs in invalid trials compared to the neutral condition. And we and other groups have now replicated these bidirectional attentional trade-offs using 3 targets, using an estimation task where observers report the precise target orientation at different locations across the visual field. And other groups have found that this also occurs across the full contrast response function in the form of response gain. So these temporal attentional trade-offs are quite robust. So next we ask, how long do these temporal attentional trade-offs last in time? And to address this question, we varied the interval between the two targets. We call this interval the stimulus onset asynchrony, or SOA. And we measured the effect of temporal attention at each SOA. So the top row here shows perceptual sensitivity, D prime, as a function of SOA on the x-axis. which again shows better performance for valid trials in blue versus invalid trials in red for both T1 and T2. But you can see there are some dynamics to this behavioral effect. So in the bottom row, I'm plotting the attentional cueing effect, valid minus invalid, as a function of SOA. And you can see that the perceptual trade-offs peak when the stimuli are separated by about 250 milliseconds for both targets, but disappear at both shorter and longer SOAs. We have now developed a computational model, the normalization model of dynamic attention, that accounts for these data. The solid lines show the model fit to the data points. And I won't say any more about this model in this talk, but I'm very happy to discuss it more in the Q&A afterwards if anyone is interested. Now, the data and the model both indicate that whatever neural constraint is responsible for these perceptual trade-offs, it has a time scale on the order of a few hundred milliseconds. And this is an important time scale for much of human cognition and behavior. You know, a lot of our cognition and behavior kind of moves at that hundreds of milliseconds time scale. But the neural mechanisms underlying these temporal attentional trade-offs, these temporal constraints, are not yet understood. So here, in the studies I'm going to talk about for the rest of today, we investigated these neural mechanisms. We first asked, how do we attend to anticipated points in time? And, you know, why can't we just sustain attention and perception across time? What is the source of these temporal constraints that lead to these attentional trade-offs across time? So to uncover the neural mechanisms of temporal attention, we need to isolate temporal attention from the related but distinct concept of temporal expectation. So expectation is based on predictability. What is the likelihood of something happening at a particular moment in time? Whereas attention is based on relevance. How important is that moment for my current task? So for example, let's say you pass by the Red Lobster every day on your route to work. So you can predict when it's going to appear. But, you know, you've never gone to Red Lobster. You never intend to go to Red Lobster. So the relevance of that moment is low. On the other hand, the moment when you pass the blue van is both predictable because you can see it coming and relevant since it's in your lane of traffic and you need to avoid it. So for any given time point, we can quantify the probability that any stimulus will appear, that's timing predictability, and the separate probability that a task-relevant stimulus will appear, so that's this time point relevance. Now, in the time domain, predictability and relevance often vary together. So a relevant stimulus like the blue van might appear with more or less predictable timing. And most of what we know about the neural mechanisms of temporal attention comes from studies that have manipulated these two variables in tandem. So to isolate temporal attention from temporal expectation, we need to manipulate relevance while controlling predictability, like comparing the predictable appearance of the blue van to the predictable appearance of the Red Lobster. And this comparison is exactly what the two-target temporal cueing task achieves. Time points that are perfectly predictable, but vary in relevance from trial to trial according to the pre-cue. So we use this approach to isolate the neural mechanisms of temporal attention from those involved in temporal expectation. And we investigated the underlying dynamic mechanisms using the high temporal resolution method of MEG. So in two matched studies, we asked first, how does the brain prepare to select a task-relevant stimulus at a specific moment? And second, how does temporal attention affect the representational dynamics of a selected stimulus after it appears? So the analyses for this first project were led by a graduate student in my lab, Karen Tian. And we used here the same two-target temporal cueing task that I described before to manipulate temporal attention while controlling temporal expectation. Here, our goal was to uncover any anticipatory top-down modulations of visual cortex leading up to the target times. And to do so, we drove the visual cortex continuously using a flickering noise stimulus, and then measured the steady-state visual evoked response, or SSVER, in a time-resolved fashion throughout the trial. The grading targets were embedded in this flickering stimulus. So here's what this looked like slowed down. So we have the stimulus is going to flicker, 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 and then there's a target, another target, and flicker, flicker, flicker. Okay, so behaviorally, then we confirmed that the attention manipulation worked with higher tilt discrimination performance for valid versus invalid trials, allowing us to then investigate the neural mechanisms. So the SSVER probe that we're continuously presenting throughout the trial, this flickering probe, allows us to get this continuous time-resolved tracking of visual cortical modulations during the anticipatory target period. So to quantify these visual cortical responses during this period, we measured the intertrial phase coherence, or ITPC, from visually responsive channels. So ITPC is defined as the phase consistency of this periodic SSVER signal across trials. So a neural response that is better driven by the stimulus will be more phase locked with the flicker across trials and have higher ITPC at the stimulus frequency. So for example, if this is the stimulus flicker, like this, flicker, flicker, across trials, if the visual response follows that flicker very well, then we're going to measure high ITPC, high phase consistency. If the visual response doesn't really follow the flicker and it's kind of randomly related to the flicker, then across trials, we're going to measure this low ITPC, low phase consistency. So again, this more reliable response to the stimulus will result in a higher ITPC at this stimulus frequency of 20 Hertz. Okay, so first we confirmed that there was indeed a strong ITPC signal at the SSVER frequency of 20 Hertz in visually responsive channels, which were located in these posterior regions as expected. So here I'm plotting the frequency spectrum frequency across time, and you can see this very strong ITPC response at 20 Hertz throughout the SSVER stimulus. And now this allows us to investigate how this stimulus-specific SSVER signal is dynamically modulated during this anticipatory period, right? So we now have this time-resolved measure of the visual responsiveness of the cortex. Okay, so what are these anticipatory modulations of the visual cortical responses? Here, I'm plotting ITPC on the y-axis at the 20 Hertz stimulus frequency across all trials as a function of time on the x-axis, with the anticipatory period between the pre-Q and T1 indicated by this gray bar. So here the trial events are in these vertical lines. So we have the pre-Q, T1, T2, and the response Q. And we're interested in what happens during this anticipatory period between the pre-Q, the time when the participants receive this cue to get ready to attend to T1 or T2, and the appearance of T1. So our first observation was that ITPC steadily increased during this anticipatory period, an overall effect of temporal expectation of the upcoming predictably timed targets. And we characterized this ramping behavior using a linear fit to the data and found a positive slope across observers. So this ITPC signal reflecting some form of top-down modulation is ramping up gradually over the course of this anticipatory period. We also found evidence for periodic modulations of ITPC during this anticipatory period, which were most prominent at delta frequencies around 2 Hertz. So this delta band modulation was consistent across both pre-Q conditions separately. And here I'm showing there's a visualization of the fit of a delta band modulation to this data. And you can see that it captures some extra variation in this data compared to the linear fit. So these findings are consistent with previous work showing that ramping and delta band modulations, though these periodic modulations reported previously have usually been reported in the context of having a rhythmic stimulus at the same frequency, whereas here we know that they're endogenous. Okay, so now what is the effect of temporal attention over and above temporal expectation? So we found that temporal attention shifted the phase of this delta modulation of visual cortical responses by about half a cycle. So here is the ITPC time series again, but now split between by attention condition with pre-QT1 in blue and pre-QT2 in red. And we can see that these time series are roughly in antiphase. We quantified the phase shift for individual observers, which is shown in this gray circular histogram. So a little gray bar here represents one individual observer, and the black line is the mean, and the arc here is 95% confidence interval. And we can see that there's this, again, near half a cycle phase shift across observers with an average phase shift corresponding to 218 milliseconds. And this value is interesting because it's similar to the 300 millisecond interval between the two targets. It's also similar to the 250 millisecond interval that leads to maximum temporal attentional trade-offs that we have measured behaviorally. Okay, so in summary, we used SSVER to continuously probe visual cortical responsiveness before anticipated stimuli. And we found these anticipatory signatures of temporal attention and expectation. First, we found that there's temporal expectation of predictable targets was associated with, first, a slow ramping increase in visual cortical responses. And second, this periodic modulation of visual cortical responses in the delta band. Over and above this modulation by temporal expectation, temporal attention shifted the phase of this delta modulation by about half a cycle. And we propose that this may mediate these trade-offs between sequential stimuli that we observe behaviorally.
Rachel, can I ask a question?
Oh, sure.
I just want to ask the measurements in when you're talking about the measurements that you showed from the brain activity, is it from posterior electrodes that are near visual cortex? It's not from the whole brain, right? It's only from occipital or occipitotemporal?
That's right. So what we did was for each session of this experiment, we selected the 5 MEG channels that showed the strongest response to the SSVER stimulus.
Okay.
And these were, as we might expect, located in posterior areas.
Okay. Okay, brilliant. Thank you.
Yeah, thanks. Okay. Okay, so what I just showed you was these anticipatory neural correlates of temporal attention, how the brain is getting ready to attend to one target at a precise moment in time. And in the next study, I want to talk about, we're moving from this pre-target period to the post-target period, now asking, how does temporal attention affect the representational dynamics of a selected stimulus? And this study was led by Jiating Zhu, another graduate student in my lab, along with Karen Tan. Okay, so the goal of this study was to measure the impact of temporal attention on the time-varying representations of each stimulus after it appears. So here we use the same two-target temporal cueing task that we've seen a couple times already, and now without SSVER, so no more flicker. And this is to obtain clean measures of the neural responses to each stimulus. So once again, we confirmed that temporal attention improved orientation discrimination performance with higher perceptual sensitivity for valid versus invalid trials. So now let's look at the neural data. Okay, so first we just looked at the evoked responses to each target in visually responsive channels. So here I'm plotting the amplitude of that MEG signal, just kind of like an ERP, but for MEG. This is from, again, these top 5 visually responsive channels selected separately from each subject, each session. And I'm showing pre-QT1 in blue, pre-QT2 in red. And again, the vertical lines show the events of the trial. So this is the onset of T1, and this is the onset of T2. And there's a clear evoked response to each target, as you can see, but no difference between pre-QT1 trials in blue and pre-QT2 trials in red. So these lines are really overlapping. And indeed, we found no effect of the pre-Q on the evoked response amplitude for any number of channels that we selected. So here on the x-axis, I'm showing the number of channels we selected in a similar way. So how responsive were the channels to each stimulus? We selected, the top 1 channel, the top 2, the top 3, so on and so forth, and just never found any difference in the amplitude of T1 or T2, or in the latency of any of these evoked responses. So here we really found no evidence that temporal attention changes the visual evoked response when temporal expectation is controlled, right? So when the stimuli are perfectly predictable and the only thing that's varying is the relevance of each time point, at least in our hands here, we saw no evidence for changes in the just raw evoked responses. Okay, so next, we use decoding to examine stimulus-specific representations and how they unfold over time. Now, importantly, in this task, the stimuli were independently tilted around the vertical or the horizontal axes. So when they were tilted, the subjects are always performing the same task, which is this clockwise or counterclockwise discrimination. But the targets can be tilted either a little clockwise or counterclockwise around vertical, or they can be tilted a little clockwise or counterclockwise around horizontal. And these tilts are quite small, maybe two degrees of tilt. They're at the individual observer's threshold. So this design means that we can decode vertical versus horizontal orientation as a measure of the stimulus representation, and that the stimulus axis was orthogonal to the participants' counterclockwise versus clockwise response, allowing us to isolate the sensory features of the stimulus in our decoding from the participants, any brain activity related to the participant's decision, the participant's response, the participant's motor action, etc. So now, as expected from previous work, we found robust decoding of both targets. So here I'm plotting decoding accuracy for T1 on the left and T2 on the right across time. And you can see there's a very clear peak in decoding about 150 milliseconds after the target onset, and then decoding slowly falls off, and that's quite consistent for both targets. Similar peak levels of decoding for the 2 targets. Okay, So this is good. So we can decode the target orientation. So now, what is the effect of temporal attention on this stimulus representation? And what we found is that temporal attention enhanced the representation of the first target, T1, at about 250 milliseconds after it appeared. So here I'm plotting T1 decoding accuracy across time on the x-axis for attended trials in yellow and unattended trials in purple. And we found a significant window in which decoding accuracy was higher when T1 was attended than when it was unattended. And interestingly, this window was at an intermediate processing stage between 235 and 300 milliseconds. So not the early times that we might have expected from previous work on spatial attention or feature-based attention, but at these later kind of what we're calling an intermediate processing stage, just before the appearance of the second target. And this is later than we expected based on not only studies of spatial and feature-based attention, but also of temporal expectation, just temporal predictability alone. All of those studies have found increases in visual responses at around 100 milliseconds. We also found no increases in decoding accuracy with temporal attention for T2, suggesting some temporal asymmetry in selection mechanisms for the first versus the second target. We have also now replicated these findings in a second data set, finding attentional enhancement for T1 in approximately the same time window, no attentional enhancement for T2. So especially given that this timing of the attentional enhancement for T1 was later than we expected, we next asked, where in the brain does it come from? So from our earlier analysis of the evoked responses, we didn't see any suggestion of changes in brain activity in these early visual areas. So where is this attentional modulation really coming from? We took advantage of the possibility of source reconstruction with MEG data to answer this question by decoding from source space during the significant time window. So first, we established that the topography of the overall orientation decoding accuracy during this time window was what we would expect, with the highest decoding performance in occipital and parietal regions. We also confirmed that the time courses of decoding across regions were, again, what we would expect, with occipital and parietal regions, so occipital in purple, parietal in this dark red, peaking earliest, followed by temporal in the dark orange, and finally frontal in the light orange. So these results verified the source reconstruction methods. And next, we compared decoding performance in attended versus unattended trials. So here I'm plotting this comparison, pre-QT1 versus pre-QT2. So more red means a stronger decoding accuracy when the target was attended. So this is for the T1 representation. So we're looking at T1 decoding accuracy, attended versus unattended. More red is higher decoding accuracy for attended trials. Again, it's all within this significant time window from the channel level decoding, 235 to 300 milliseconds. To our surprise, we found that the strongest effect of temporal attention was localized to frontocingular cortex, which you can see here, where we found this significant spatial cluster that was strongly left lateralized. So here I'm showing this cluster, spatial cluster region that survived the multiple comparisons in these open circles here. So you can see it extends across the left frontal cortex, as well as going around over the top here into the medial surface into the cingulate cortex. So left frontal cingulate cortex is where we're seeing this attentional enhancement at this intermediate processing stage. And you can see it's very strongly left lateralized. Basically nothing going on in the homologous regions on the right hemisphere. Okay, this left lateralization is actually consistent with some previous studies showing that temporal attention tends to recruit more left lateralized frontoparietal regions compared to spatial attention. But it wasn't known before that stimulus information is actually being sent to these anterior regions. I think it was previously thought that these regions might be involved in attentional control, and they may well be, but we also see the presence of stimulus-specific information, this significant decoding accuracy in these regions. And you can see that the topography of the attention effect focused on these anterior regions is very different from the topography of the overall decoding performance, which is highest in posterior regions. Okay, so to summarize this part, we found that temporal attention also impacts the spatiotemporal dynamics of neural representations of attended targets after they appear. And here we found that there was enhanced representation of the first target's orientation at an intermediate processing stage around 250 milliseconds, just before the arrival of the second target. This enhancement was localized to the left frontocingulate cortex, overlapping with the cingulo-opercular network, which is an interesting network in that it has been proposed to, among other things, select and reformat internal representations for action. It's also been found to affiliate flexibly with other networks according to task demands. So one possible interpretation of these results is that temporal attention may send target information to frontocingulate cortex to protect it from interference in visual cortex by the subsequent stimulus, T2, which may explain why we see these attentional enhancements only for T1, which has the potential to be overwritten in just a few hundred milliseconds by T2, but not for T2, whose representation could potentially linger as long as it needs to in visual cortex to be read out by subsequent decision mechanisms. Okay, so overall,
Rachel.
Oh yes, hi.
Hi. So I can ask a quick question. Hi.
Yeah.
So does it mean that this effect you're seeing, is it some sort of shielding mechanism that is related to the appearance of T2? So if T2 would appear after 200 milliseconds, you will see it after 150 milliseconds?
Yes, it's a very good prediction. I think that is indeed what this account would predict. Like if T2 were coming sooner, you might see this transfer of information.
Yeah, whatever. I use the word shielding not to refer to
shielding exactly. That's the idea that we need to protect this representation from the onset of the next stimulus in visual cortex. Visual cortex cannot protect it because things just come into visual cortex and are immediately overwritten by the next thing that comes in. So where can we stash this representation, if you will, in order that we can preserve it to do, the more flexible decision readout that we will need to do in order to assess whether it's a clockwise or counterclockwise target. That's the hypothesis. And so exactly, I think it would predict that if you had, a shorter time, that this should always happen before the onset of T2, if it were possible to send it there fast enough, right? So it's actually very interesting that behaviorally, we see these trade-offs, behavioral effects of temporal attention peaking around this time interval, around this 250 millisecond, 300 millisecond time interval. At the shorter time intervals, around 100 milliseconds, actually T1 is strongly backward masked, and performance for T1 is quite quite poor. And so it may be that at very short time intervals, there's just not enough time to deal with T1, to send it somewhere else before it gets quickly overwritten. But at these longer time intervals now, you can ship this information somewhere else in the brain where you can protect it. This is the idea. Of course, we need to test this, so it makes many predictions for some future experiments. Yeah.
Okay, thanks. Yeah.
Great. Okay. Okay, so I showed you that in several behavioral studies, we first of all found evidence for the selectivity of temporal attention. So voluntary temporal attention trades off its effects on perception across time. Then in two MEG studies, we isolated the neural mechanisms of voluntary temporal attention from those of temporal expectation and found two new mechanisms associated with temporal attention. So first of all, in anticipation of a predictable stimulus, visual cortical responses are modulated periodically at a delta frequency, which shifts in phase depending on the timing of attention. And second, temporally attended stimulus information is routed to frontocingulate cortex before the onset of a temporal competitor. So at a behavioral level, then, we see that there are these common behavioral trade-offs across temporal and other types of attention. You attend to one stimulus, your performance improves for that stimulus, but is impaired for a competitor stimulus, whether that other stimulus is separated in space or in time. At the level of the brain, though, we find that there are these specialized neural mechanisms, at least some specialized mechanisms for temporal attentional selection that may reflect the demands of dynamic sequential processing. So going back to this three-pronged approach, it's still early days here, but here are how the results I just showed you fit into our developing understanding of dynamic attention, and particularly the selective aspects of temporal attention. So we see at the behavioral level, these perceptual trade-offs. We also see in studies I didn't tell you about today, some ocular motor trade-offs, so changes in the behavior of microsaccades as we approach the timing of predictable and then attended targets. We also, I briefly touched on this normalization model of dynamic attention, something we're continuing to develop, which can predict these perceptual trade-offs across time and also the time course of these trade-offs, and really helps us to formalize our theories of what might be going on in the brain and develop new experiments to test them. And then finally, what I focused on today was this new work where we're now delving into the neural mechanisms of this, where we've so far found these changes in visual cortical responses and leading up to anticipated temporally attended stimuli, and also changes in stimulus representations following these stimuli. And really, the results of both of these neural studies, this phase shift of the anticipatory delta oscillations, this frontocingulate improvement in decoding performance, none of these were what we expected when we went into these projects. So it's been very interesting, and we're continuing to figure out what's going on here. A big question that we're actively pursuing in my lab now is, what are the neural and computational sources of the temporal limits that we observe in behavior? And altogether, this work helps us move toward a more mechanistic understanding of how perception and attention unfold across time. So we can do something like this, bike through these busy streets, despite the real temporal constraints on visual processing. So I'd like to just end by thanking the people who contributed to these projects in bold, thanks to Karen Tian and Jiating Zhu, graduate students who worked on the MEG projects, Marisa Carrasco, David Heeker at NYU, my postdoctoral advisors there. We started this whole line of research together there, as well as Antonio Fernandez, who I did not bold, but also contributed to some of the behavioral studies I showed. And of course, thanks to funding sources, and thanks so much to all of you for tuning in.
Rachel, thank you so much. This was really a great talk, and I want to invite everybody to unmute yourself and give Rachel a big applause for a great talk. Thank you so much. And I already see that there is a question. Are you happy to take questions?
Oh, yes, absolutely.
Okay, wonderful. So Igor, we'll start with you.
Thank you. Thank you, Rachel. Great talk. And I was wondering if this voluntary temporal attention has to do with attentional blink. Do you think the mechanism is there, or it is a different process that is responsible for that?
Yes, great. Let me, just in case people may not be familiar with the attentional blink, although probably everyone may be. Let me find a little picture of that. Okay, so here we go. This is just an illustration of attentional blink task. In this task, you get a sequence of rapidly presented stimuli like letters. Let's say you have two targets you have to identify. Let's say you have to identify the red letters. Okay, and the finding of the attentional blink is that when these targets occur a certain interval apart, you have this difficulty in reporting T2 when it occurs about 200 to 500 milliseconds after T1. Right, so you have this decrement in performance of being able to report T2, and this is the attentional blink phenomenon. And so there are a few differences between the setup of these attentional blink tasks and the setup of the voluntary temporal attention task, namely that in the attentional blink task, you actually don't have any advanced timing information about when these targets are going to appear. You're just sort of waiting in a state of sustained attention until they arrive and they're defined by the feature. So they're actually defined by the feature rather than being defined temporally, being defined by the time that they appear. So this is the main difference kind of procedurally between attentional blink and the voluntary temporal attention. So, you have, so as a result, we, for example, in this recent review paper, we find it more helpful to think about attentional blink as a kind of reactive form of temporal attention, because you're sort of waiting for some triggering feature, and then you may allocate attention to get that feature instead of sort of prospectively allocating it in advance because of timing information that you have. So that's an important difference between attentional blink and the voluntary temporal attention that we're studying. That said, what's very interesting is that the time course of the attentional trade-offs that we see using voluntary temporal attention manipulation are very similar to the time course of the trade-offs that we that are commonly seen during the attentional blink. So if I can, if I have a good picture of this, I will get it for you. So let me go back to this picture. So focusing on T2 here, yeah, we see that we have this actually very kind of U-shaped function of performance for T2 when you are attending to T1 in the invalid trials. It's quite similar to the time course typically seen for attentional blink. So we think that it's quite possible that whatever temporal constraints are responsible for the attentional blink may also be at play in this voluntary temporal attention situation. And the nice thing about doing it with voluntary temporal attention is that it's in some sense much simpler, because you only have these two stimuli. Exactly, the observer knows exactly when they're going to appear. There's no distractors, there's no masks, and so forth. So you can really start to isolate the mechanisms responsible for these trade-offs per se.
Thank you.
Yeah.
Wonderful. Yeah, I really like the idea. And I also think, I'm thinking about change blindness, but it's the same idea and some enhancement of the attention of the first stimulus. I don't know, but Josh, you go. Where's Josh? I don't know. I thought Josh Strait is.
Oh, yeah.
Oh, yeah,
Yeah,
here you are. Okay, good.
Yeah, sorry, my internet connection is unstable. To what extent, Rachel, are your results robust to differently positioned targets and distractors? Also, is there anything interesting in the relationship between the distractor identity and the performance? And finally, sorry for so many questions, can you correlate performance with your brain activity across observers?
Okay, yeah, thanks. Those are all great questions. So in terms of the position of the stimuli, what we have done so far, and this is with Marisa Carrasco and Antonio Fernando, is just tested to see whether having both targets at various positions around the visual field made any difference to the size, like the magnitude of the attention effect. And we didn't see any variation across the visual field. So we saw that similar size attention effects, no matter whether the stimuli were presented vertically or, in the horizontal meridian or in the vertical meridian and so forth. What we have not done is separate the spatial position of the target, and the distractor, as it were, separate the spatial position of T1 and T2. Because we haven't done that so far because we didn't want to introduce the need for a spatial attention shift. So like if the T1 were on the left and T2 were on the right, then there may be an extra mechanism involved to shift your spatial attention from one location to the other. But it would be very interesting, I think, to look at that and to look at how the relative spatial position may affect the trade-offs, to look at how that interacts with the interval between the targets and so forth. So we haven't done it, but I think that would be quite interesting. Let's see. I think your last question was about correlation between behavior and the neural responses. And so far we've looked a bit at that, but we have not seen any tight correlation between the neural measures that we found at a group level, the neural effects at a group level, and the behavior at an individual level. And I think it's quite likely that the reason is that these experiments were not really designed for individual analysis. We have the approach of having, fewer subjects. We have just 10 subjects, but we test them a lot. So we have a lot of data from each subject, but as a result, not very many subjects in order to do these correlations. And also the effects tend to be quite consistent across subjects. So sort of not that much individual variability that we can marshal for these correlations. But I think that will be very important to, in the, going into the future, to try to get maybe larger sample sizes or do different kinds of measures that we can do trial by trial correlations, which we can't really do with either the decoding measures or the ITPC measure. Anyway, I think it's a very important direction. And sorry, Josh, can you remind me of your second question?
I just said, is there anything interesting in the relationship between distractor identity and performance?
Yeah, right. Good. Okay. Yeah. So we've started to look at this a little bit. Now we don't have we've so far really focused on these simple stimuli, these gabors, because we have some knowledge about how they're processed in the visual system, and it helps us to be able to model these basic processes and so forth. But we have looked a bit at how the features of the two targets relate to each other. So for example, if both targets are vertical, or if one target is vertical and the other target is horizontal, or if both targets are clockwise, or if they have different orientations. And there do seem to be interactions in terms of the relative feature values of the two targets, that we are able to capture sort of in a model setting, more at a decisional level. So more like if you are if the first target is clockwise and the second target is counterclockwise, you may be a bit, and then you're asked about T1, you may be a bit more likely to make a counterclockwise response because it kind of leaks over from the non-target. So there are definitely some interactions that are feature dependent. I think so far it looks like they may be more at a decisional level. For example, when we have the continuous estimation tasks, we don't really see much evidence of the response being biased toward the non-target, or really any swapping with the non-target, and so on and so forth. So that's what we have so far. But I think as we build this up and have more complex stimuli and so on, it will be interesting to look at the more at the non-target influence. Yeah, thank you.
Rachel, are you okay to continue with the questions?
Oh, sure.
Okay, lovely. So Nir has a question, and I'm after him.
Thanks, Rachel. It's a kind of question that you can say we'll talk about it at a different time. But is there any, I mean, I'm not even sure if I'm that convinced with all those recent findings I'm going to talk about, but the whole discussion about rejecting distractors based on their timing. Is this something you can account for in your model? Is this something that maybe is impossible, like based on your model at a certain time window? I'm kind of curious because this whole, relevant, but what is task relevant, right? Like distractors are relevant in one sense because they can interfere with our performance. So we need to reject them, but at the same time, they are often not relevant to our responses, for example. So the idea of task relevance gets a bit muddled in that case, but if you have a mechanism to reject in a timely manner task relevant, or sorry, to attend task relevant moments, can you also reject distracting moments?
Yeah, it's an interesting question. So yeah, so first of all, I think I would say that in terms of just defining task relevance, the way that we think about it is, will that moment improve? If you get the information in that moment, will it improve your task performance? So you could almost think about it in a kind of like optimal decision framework where some stimuli are going to be useful for completing your task, and those will be relevant, and some stimuli will just not be useful for completing your task, and those will be irrelevant. So that helps us a little bit to sort of just simplify the definition of task relevance in that case. But I know what you mean, that distractors, you might think it would be a good idea to know when they are going to happen, so you can actively, if you were able to do that, so you could actively block them. And there is some evidence, I think, for this from like the more like working memory literature. I know there's this interesting paper by Bonifant and then Jensen, where they find that if you can predict the timing of a memory distractor, you can seem to be able to adjust your alpha phase in such a way that it will suppress it, and so forth. But I think the data that we have are not perfect for really addressing the question of whether there is active suppression versus just an enhancement that then results in a relative reduction of performance for the other target through some other stage of a competitive process. And so, we have the neutral condition, of course, which is quite important, but that really just provides a kind of an intermediate, like baseline point from which to compare attended and unattended targets. We see that there are costs for unattended targets relative to the neutral condition. But again, is this due to sort of like an active suppression mechanism? I don't think, the data that we have really speak to that. And it's a tricky, I mean, as we know from other work trying to look at active suppression in visual search and so on, it's a quite a tricky question. Yeah.
Yeah, okay, yeah. I mean, I'm not sure about it as well. Just there is this recent papers that came out from I think Jan Tive's lab, where they have preparation in time, but I think it's not as refined as your definition here. And I'm not sure how to put these things together, but I think it will be interesting what happens if T2, for example, if you have the knowledge that it can, really impair your ability to make this perceptual judgment later of T1. For example, if it's, I don't know, if it's close in terms of its orientation or something like that, that makes it not only irrelevant, but actually something you don't want to look at, so to say.
Right. Yeah, it is actually quite interesting. Actually, one thing that's I'll kind of bring back here is the finding of this sort of antiphase relationship in the delta modulation. It is something that originally we weren't expecting, because we thought, oh, in order to enhance a target, you can just ramp up and maybe get higher by the time that target appears. But here we find this kind of antiphase relationship. And once we saw this, as we were thinking about it, we thought, maybe what's happening is when you need to select a target at a very precise moment, it's actually better to do it with an oscillatory mechanism, because what this does is sort of simultaneously should enhance the one that you're attending to and also relatively suppress the one that you're not attending.
That's interesting. Yeah.
You know what I mean? So anyway, this is also, we need to follow up on that, but that's one thing that we're thinking about, of whether the oscillatory mechanism may actually be particularly useful in a context where there are temporal competitors, where there are distractors effectively.
Thanks.
Rachel, I actually have a few questions if you're, but you don't have to answer all of them. I just want to comment first on this. So because it seems to me that this can be an antagonistic way, but also I'm wondering if there's maybe some phase shift that is corresponding to the delta between T1 and T2. I mean, if you ignore the dip, you can actually, I mean, it may seem that the blue one, which like the pre-Q of the T1 is actually preceding the activity of the measurements of the orange one of the T2. And I wonder if you do some autocorrelation, if you see that if you see the peak of the fit that corresponds to the delta between the T1 and T2 offsets. Just an idea.
Yeah, so are you saying looking at the interval between T1 and T2, which is about 300 milliseconds?
Yeah,
like,
would that
does
be
it relate to the phase shift?
Yeah, I'm just wondering if
Oh
there's
yeah, that's right. Here, I show you this. So the phase shift corresponds, this phase shift, which is like 170 something degrees, corresponds to 218 milliseconds. So it's similar, but it's not it's not exactly 100 milliseconds. It's in the ballpark. It's also in the ballpark of kind of the maximum behavioral trade-off interval, 250 milliseconds. So actually, one thing we are starting right now experiments to look at is, okay, if we change the interval between T1 and T2, does this frequency and this phase shift stay where it is? And maybe, that's what the brain can do. And that's why we observe the trade-offs at this time scale. Or alternatively, will the frequency and the phase shift now flexibly adjust to match the interval between T1 and T2? Like, let's say if they're farther apart, is the frequency going to get slower and the phase shift get longer? Yeah.
Yeah, brilliant. And I also wanted to ask, but that's more of a philosophical question. It's not, but still, I mean, this is done when people are actually sitting down and being like not walking around the environment and being active. And I'm just wondering whether you think that the actual movement in the environment may qualitatively change these, and maybe, for example, there's more attention in general directed to temporal changes when we're moving around versus when we're actually sitting down. And the most of the changes that can come when you're doing an experiment is when you're from the computer that you're looking at, but it's no threat to you more, most of the time. So I'm wondering if you think that this may, I mean, have you considered, measuring things while people are moving, or maybe in some more realistic environments like the one you showed on the bike, which is, amazingly complex and maybe unsupervised or uncontrolled, but and it's not that we're in life risk, every on a Manhattan street on a bike every day and every time, but still it's another extreme of, lab versus lab, protected, controlled environments. So I'm just wondering about that.
Definitely. So I think a lot of the temporal information that we have in natural behavior about when something is going to become task relevant is actually due to our own bodily movements through the world and our own eye movements as well. So, for example, here's two examples that come to mind. Like, let's say you're crossing the street, right? If it's let's say it's an relatively empty street, you can kind of zone out, right? You can kind of zone out while you're just walking across the street, but then there's going to be some moment when you're approaching the curb. Let's say there's like a little curb on the other side of the street where you have to now, attend to that curb at that precise moment that you need to prepare to step up on the curb, right? And so in this case, your temporal attention is going to be linked to your, motion, your self motion across the street, or even in, really simple everyday actions, like you need to pick up a cup, a cup of coffee or something, right? Like you're getting ready, you're like, I want to take a drink. And so you get your body ready to move, and then you allocate the attention at that moment to the cup right in time so you can get the information to your motor system to grasp the cup. So I think a lot of our our dynamic allocation of attention in natural behavior is tied to our movements and our goals that are kind of like internally generated by us, right? Like not that's like instructed by an experimenter, but that we're like, I want to do this thing in the world. Therefore, I need to get this information. I need to get it now. If I get it later, it's not going to help me. I need it now, before I make my action. So I think that's a large source of the dynamic variation in attention. And I think it's very important, to move in that direction to try to introduce some of this kind of perception action cycle into this study of attentional dynamics. So far we've really started so simple, as simplest as we possibly can. And of course, it's a research strategy to simplify as much as you can, to use as simple stimuli as you possibly can, that you can have a chance to understand and model and leverage our knowledge of this simple situation for starting to get a handle on the neural mechanisms and so forth. But I think, yeah, I mean, as we get some clarity about what's going on at this very basic level using these simple things that we can model, that we can manipulate, and that we can measure with neural measures and behavioral measures, psychophysics. I really hope we can slowly build up step by step to these more complex scenarios, getting eventually right. Like the goal really is in the end of the day to be able to understand how your attention is going to be dynamically allocated during your Manhattan bike ride. So, lots of work to be done.
Yeah, beautiful data. Thank you so much. And I just want to ask, because I see that Josh has raised his hand again, and I don't know if that's really meaningful or by mistake. So Josh, if you're still there, is it the case that you want to ask something or follow up?
Yeah, sorry, Mike's here too. Michael Morgan asks Rachel whether it's not a follow-up. Michael Morgan asks whether you'd like to comment on the doctrine of prior entry, which says that attended stimuli are perceived before unattended stimuli.
Yeah. So that so I guess prior entry is more of a kind of a time perception situation, right? When you're trying to estimate the timing of different events. So this is I would put this more in the category of time perception, like so we can estimate when something happened or how long something lasted and so forth. But what I think it is an interesting question of how temporal attention might interact with these notions of with time perception. I don't know of any work that comes to my mind that's looking at how temporal attention affects time perception. There is work on how other kinds of attention affect time perception, like work where if you're attending to something, there's like this temporal dilation where you perceive it as longer, and so on. And I'm trying to remember if there's work on like spatial attention and perception of like timing onset. I think so, yes.
There's stuff about hazard function that Domenica Buetti has been doing. And representation in V1 and other areas, I think also with Vincent Walsh, but I can look it up and send you some papers if you're interested in this direction.
Sure. Thanks. Yeah, I don't know, does that answer Mike's question?
I'm not sure.
Okay, thanks, great.
Okay, so thank you so much for joining us, Rachel. It was a really fascinating, interesting, and thought-provoking ideas and data. And again, thank you so much for this wonderful talk. Next week, we're going to have Avital Hahami from UCL here with us. So again, thank you, Rachel, and thank you everybody else for joining. And yeah. And see you next week.
Okay, well, thank you so much. It was a pleasure, and thanks everyone for your questions as well.
Lovely. Our pleasure. Thank you.
Thanks, Rachel. Bye.
Bye.